Michael Snyder is the Stanford W. Ascherman Professor of Genetics at Stanford University and has co-founded 17 companies, including Personalis, Qbio, and January AI. For GenomeWeb's 25th anniversary, 360DX managing editor Tony Fong spoke with him about the impact of genomics and the future of the field.
Walk us through a few notable developments over the past 25 years in the genomics field and why you think they're notable.
Well, I guess in 2000, we only had a draft of a genome that was, done by slugging it out with old Sanger kind of technologies. And obviously, there's been a revolution since then. All these next generation technologies for sequencing genomes, the first version came out was 454, and that still took time to sequence. Then along came Illumina, and now a whole host of other technologies. So it's at the point where you can at least generate the sequence data within a day and across many genomes actually now in a single run. It still takes a little time to analyze it. But the bottom line is if you have to push it within a few days, you can get someone's genome sequence. And sometimes that is useful. So I would argue the other big areas, bringing genomics into diagnostics was huge, both for cancer field. Now, some areas of cancer, like lung cancer, if you don't get a genetic or genomic test, I would call it malpractice. You really should be getting genomic information because it guides the treatments you would do. Likewise, for solving these undiagnosed disease for newborn kids, there's been a revolution there, where now about 40% of these mysterious cases you can solve through genome sequencing. Then there's many other omes, as you pointed out. There's transcriptome, proteome, all these fields have emerged. We can now dissect and analyze those in incredible detail. Some of the latest things are we can now do single cell proteomics. We went from single cell transcriptomics for the most part, then later single cell genomics and methylome, and now you can even do single cell proteomics, which is pretty powerful and get reasonably deep. So I think our resolution of what we can measure and then also how we impact all the applications have just been enormous, both for the research and the clinical side.
Moving on to precision medicine, and you touched upon this a little bit in terms of cancer, the use of precision medicine in cancer. What do you think needs to happen in that field before we can really tailor diagnoses and treatments to individual patients on a wide scale?
Well, again, on cancer, we can do it in certain areas, and it's pretty essential. These days, most people classify cancer as based on the oncogene that got mutated that's getting expressed, which you discover through genomics. So I think there's been quite a bit going on there in the cardiovascular disease. There's a suite of a few hundred proteins everybody zooms in on when they're trying to look for genetic basis of defects. And I think I wouldn't say that's necessarily affected the treatments a whole lot, but it sure helps when you know exactly what's wrong, that you really are on the right track for drugs.
We would be amiss if we didn't mention what's happening in Washington, D.C. What are some of the worries you have about what is going on there in terms of genomics-related research, with the cuts in funding and layoffs happening, and the shutting down of whole facilities?
All the NIH officials I know work really, really hard already before the cuts. And so this has been pretty dramatic. I think it's been morally devastating for the folks I've talked to. And a lot of it's not only that they've lost some of their friends who they've worked with who are quite competent, but also I would say they've, there's a lot of uncertainty and lack of appreciation. NIH is a crown jewel of American science. I mean, I think the rest of the world's envious of American science. We put a lot. Here's one way to look at it. NIH is the R&D engine, if you will, for a $4.5 trillion industry. And so we basically, the discoveries from NIH, they translate into things that not only save people's lives, but they actually have an incredible return on investment. Some people, the most conservative, say there's a fourfold return on investment. So it's totally illogical to be cutting NIH.
Where do you see the genomics and proteomics and the personalized medicine fields heading in the next 25 years?
Oh, things will be orders of magnitude cheaper. I hope we do genomes and proteomes, for $5 or less, right in the doctor's office, so we can get deep profiling. And I think the algorithms for integrating data and getting understanding will be a lot higher. From the healthcare side, we work very hard on home monitoring, meaning you could do a lot of these measurements at home, or at least the samplings. So we now can take, for example, in our lab, little drops of blood. Someone can take them, mail them to us, and we'll measure 7,000 molecules in that blood. And I think that kind of thing is going to be routine in the future.